Genetics Matters, Episode 10, Details of DNA Replication. Amazon and UPS should be jealous. Welcome back. We have seen the evidence for DNA as a genetic material. We have seen how model building based on the work of others led to fame for Watson and Crick. And we have seen many of the implications of the DNA double helix, including the semi-conservative nature of DNA replication. Today we will look at a few more details of DNA replication. Number 1. The two DNA strands in a double helix are complementary to each other, but they are running in opposite directions. Nucleotide residues in a DNA strand are linked together through a sugar phosphate backbone. Each nucleotide residue is composed of a 5-carbon sugar called deoxyribose, the D in DNA, a base, what makes A, C, G, and T different, and a negatively charged phosphate group. The phosphate group on the so-called 5' carbon of the sugar of a given nucleotide is strongly linked to the hydroxyl group on the so-called 3' carbon of the sugar on the previous nucleotide in the strand. This happens over and over and over again. This means that one end of a DNA strand has a free 5' phosphate group, and the other end has a free 3' hydroxyl group. When Watson and Crick built their model of the double helix, the only way they could keep the helix a uniform diameter and have the complementary nucleotides on the two strands interact through weak bonding was to have one strand run 5' to 3' from the floor to the ceiling, and the other strand run 5' to 3' from the ceiling to the floor. The two strands are said to be anti-parallel to each other. Usually, when we see a single strand of DNA in a figure in a paper or a book, it is shown going 5' to 3' from left to right. That is done solely to make it easy on us as left to right readers. If both strands of a DNA helix are shown, we can see their true anti-parallel nature. This has big implications on DNA replication. Number 2. In cells, new DNA strands are always, triple underline always made in a 5' to 3' manner. That means that the two new strands being made during DNA replication are being made going in opposite directions. Number 3. DNA replication starts at very specific sequences within a DNA molecule called origins of replications, or ORIs for short. ORI sequences are recognized by specific proteins that recruit the replication machinery. Number 4. Once an ORI sequence is recognized, enzymes called DNA helicases expend energy to open up the DNA double helix. These enzymes break the weak bonds between the two strands and separate them. Specific single-stranded DNA binding proteins can then keep the strands apart. Unwinding of DNA at the ORI by helicases puts positive torsional stress on the DNA on either side of the ORI. Imagine two friends holding two ropes twisted around each other to mimic a double helix, and you grab the twisted ropes in the middle and open up a bubble. Your two friends will feel the pressure grow on each end. Two types of enzymes called topoisomerases, or shape changers, can deal with torsional stress by nicking one or both DNA strands. Type 1 topoisomerases relieve positive torsional stress by nicking one DNA strand and allowing the other strand to pass through the nick before sealing it. Type 2 topoisomerases, sometimes called DNA gyrases, head off positive torsional stress by introducing negative torsional stress. These enzymes nick both strands of the double helix and allow the entire double helix to pass through the nicks before sealing them. Overall, the DNA gets opened up without creating knots elsewhere. Number 5. DNA replication cannot start from scratch. The enzymes that catalyze the making of new DNA strands need something to start from. We call it a primer. In cells, primers are short stretches of RNA made by the enzyme RNA polymerase, and these short stretches are made in the 5' to 3' direction. Number 6. Enzymes called DNA polymerases catalyze the making of new DNA strands in a 5' to 3' direction. DNA polymerases add DNA nucleotides, specifically called deoxyribonucleotides, onto the 3' end of the short RNA made previously by RNA polymerase. This activity in DNA polymerases is called the 5' to 3' polymerase activity. Now, DNA polymerases do not know, quote unquote, specific nucleotides. Rather, the shape of their active site works best on the shape of a proper base pair. In a DNA double helix, that shape is shared by an A nucleotide across from a T nucleotide, or a G nucleotide across from a C nucleotide. DNA polymerases rarely make mistakes, about once every 200,000 to 2 million base pairs. Actually, the error rate is even lower than that, because most but not all DNA polymerases check the last base added before moving on to the next position. This checking mechanism, called proofreading, allows the enzyme to double-check the shape of the last base pair. If it still looks okay, DNA polymerases move on. If not, the enzyme cuts off the last base added and tries again. This activity is called a 3' to 5' exonuclease activity. Proofreading adds another 40 to 200-fold of fidelity, meaning that overall, proofreading DNA polymerases make mistakes about once every 8 million to 400 million base pairs. Companies like Amazon or UPS would love to have error rates that low in their shipping and handling of packages. Number 7. As the DNA helix is opened up into a bubble, at one end of the bubble, the top old strand will be opening up in a 3' to 5' direction, meaning that the new DNA strand can be made continuously in the 5' to 3' direction using the old strand as a template. The bottom old strand at that same end of the bubble can also allow DNA to be made in the 5' to 3' direction, going in the opposite direction. But as the bubble opens up more, the newly exposed DNA strand is behind the replication machinery. The only way to get this newly exposed DNA replicated is to start over with a new RNA primer and then DNA synthesis catalyzed by DNA polymerase. In this case, the new DNA strand is made in pieces. It is discontinuous. At the opposite end of the bubble, it will be the bottom old strand that can serve as a continuous template for DNA replication. The top old strand at this end of the bubble will act as a template for discontinuous replication. This is really hard to see in your mind's eye, so draw it out on paper and you'll see what I mean. Number 8. When a DNA polymerase making a new strand meets up with another section of newly made DNA laid down previously, another activity found in a few DNA polymerases takes over. A 5' to 3' exonuclease, meaning chewing at the ends, allows the DNA polymerase to trim off the RNA primer on the DNA in front of it as it fills in the DNA strand it is working on. Once the RNA primer is trimmed off, the DNA polymerase finishes up its DNA strand and stops, leaving just one last nick to be sealed. Number 9. DNA polymerases add nucleotides onto the 3' end of DNA strands. they do not just seal nicks between adjacent nucleotides. That last function is different, and it is done by enzymes called DNA ligases. To ligate means to sew up. These DNA ligases use energy from ATP hydrolysis to attach the 5' phosphate of one nucleotide at the nick to the 3' hydroxyl of the nucleotide on the opposite side of the nick. In this way, complete DNA strands are made. Wow, that is a lot of different jobs that must be done just to get one DNA double helix replicated into 2 double helices. Even more impressive that it is done with such a low error rate. It is downright incredible when we learn that DNA replication can move at speeds of up to 1,000 bases added per second. Per second. Now, if you really want to blow your mind, check out the animation of how all this is put together at www.dnalc.org/resources/3D/mechanism-of-advanced.html. Thanks for listening, and I'll see you next time on Genetics Matters.